Although wild type lambda phages, they can be used as a cloning vector, but because of some characteristic feature, it is not so good to transform foreign DNA into the host organism. The reason is that it contains multiple restriction sites for different type of endonucleases. If genomic DNA of the lambda phage, it will contain different restriction sites for the same enzyme, then it can cause the fragmentation of the lambda genome. And because of this reason, it is not valuable for gene cloning procedures. Under natural conditions, we have the idea that transducing phage particles are produced that can carry the some part of the host genome. And then it can be transferred to another host organism by a process which is called as transduction. So naturally, lambda phages have the ability to be used as a cloning vectors. But nowadays, to solve the problem of multiple cloning sites, researchers have developed some modified lambda phages that can be used as a cloning vector. These modified lambda phages have one or two restriction sites where the DNA fragments either can be removed or inserted. A few examples of the modified lambda phages are Chiron vectors that included Chiron 4A, where the restriction sites may be 2 to 3, and the non-essential DNA fragments, it can be removed and replaced with the foreign DNA. Because here we are removing the non-essential region and replacing it with foreign DNA, तो इस वजह से इसको replacement vectors भी कहा जाता है. Like Chiron 4A, ये इनको replaceable vectors भी कहा जाता है. इसी तरह से एक और टाइप है Chiron 16, जिसमें एक ही restriction site होती है और इसको EcoR1 से कट करके तो फॉरेन डीएनए इंसर्ट कर दिया जाता है. क्योंकि यहाँ हम फॉरेन डीएनए को इंसर्ट कर रहे हैं, इस वजह से इनको इंसर्शनल वेक्टर्स कहा जाता है. सो द मॉडिफाइड लैम्डा फेजेस, they are produced by removing the multiple cloning sites by different mutational process that may be included like the substitution of bases or addition or deletion of certain bases, so that it may contain a few cloning sites. In addition to that, modified lambda phages, they also contained an extra reporter gene which is called as beta-galactosidase. And this production of beta-galactosidase after the insertion of foreign DNA, it will indicate that whether the foreign DNA it is inserted or not. For example, if the beta-galactosidase expression it is lost because of the insertion of foreign DNA within this gene, then this will indicate that okay, no transformants have been produced. Now briefly we can focus that what type of lambda derivatives are there. First example, it is the wild type lambda here. It indicates the different regions of the lambda genome. The 5' ends, that is red portions, they are indicated by the cohesive sites or cos site, where complementary base pairing is there, by which after inserting into the host organism, these complementary base pairs they can join, and the linear duplex DNA molecule it is converted into circular form. On the left side, some genes that encode for head and tail proteins are there, that is capsid genes, and at the right side also some essential genes are there. But the DNA segment between the J and N gene, as I told you earlier, it can be replaced with foreign DNA. So that's why it is also called as a replaceable region. But because of the presence of multiple cloning sites. This DNA, it is not valuable as a cloning vector. For example, here different restriction sites, they are present for EcoR1. For example, here 5 EcoR1 sites have been indicated. So because of this reason, lambda genome that is wild type, it is not well suited as a cloning vector. Because of this reason, modified lambda phages have been synthesized that are called as modified lambda phages derivative. So one of the example is Caron 4A. So here 2 or 3 restriction sites they are present for EcoR1, and here the non-essential DNA fragment after digestion with EcoR1 it can be removed, and the foreign DNA it can be inserted within beta Gal gene. Once it is inserted, the expression it is lost that will indicate the presence of foreign gene. And because here the non-essential fragment it is removed, because of this reason it is called as replacement vector. The third type it is called as Caron 16. It contains only one restriction site within the gene of beta Gal, and here the DNA fragment it is inserted. When the DNA fragment is inserted, its expression is lost, and it is called as insertional vector because we are inserting the foreign DNA but not removing any part of the Caron 16. So that's why it is called as insertional vector. Now we can say that modified lambda phages like Caron 4A and 16, they can be effectively used as a cloning vector. And here the multiple cloning sites have been removed by either substitution, addition or deletion. In addition to that, it also contains a reporter gene which is called as beta galactosidase that encode this enzyme, and if this enzyme it is produced after the insertion of foreign DNA, then we can say that the foreign DNA has not been inserted. And if the insertion is there, the expression of beta galactosidase it is inactivated, that indicates the presence of foreign DNA. If we compare these two types of vectors like 4A and Caron 16, then one can say that in case of Caron 4A, the fragment it has been removed and foreign DNA it has been inserted. So here we can clone larger DNA fragments, whereas in case of insertional vector like Caron 16, we are inserting the foreign DNA without the removal of foreign DNA, or rather without the removal of vector DNA. Then only smaller DNA fragments that may be up to 5 kilobase pair they can be cloned. So overall, because of the presence of reporter gene, these modified lambda phages they are very effective in transforming E. coli cells.